Hello everyone. Today we will start lecture 3rd of enzymes. Our today's topic is remarkable properties of enzymes. So we are going to discuss some very special properties of enzymes, which are as follows. The very first property of enzyme is specificity. That is, enzymes are very specific, or we can say enzymes are reaction specific, and for a particular reaction, a particular enzyme is needed. For example, for hydrolysis, hydroxylase catalyzes the reaction. Among hydroxylases, esterase hydrolyzes esters only. This is an example of reaction specific enzyme. On the other hand, an enzyme may be substrate specific too. Such enzymes are specific for a particular compound or a class of compounds. For instance, urease hydrolyzes only urea, phosphatases hydrolyze only phosphate ester. Lysases remove group from substrate by mechanism other than hydrolysis, leaving double bonds. So here lysase acts and leaves the double bond and a product is formed. Ligases link together two compounds. They catalyze reactions forming carbon-oxygen bond, carbon-sulfur bond, carbon-nitrogen bond, carbon-carbon bonds. Transferases transfer a group other than hydrogen between a pair of substrates S and S dash. Here S with group and S dash. S is a substrate with group and S dash is a different substrate. Then in the reaction the group is attached to the different substrate and the original substrate is removed. Many enzymes exhibit kinetic specificity. For example, esterases, although hydrolyzes all esters, but hydrolyzes different esters at different rates. Kinetic specificity means they give different rates of reaction with different enzymes and different substrates. Many enzymes are stereospecific. For instance, maltase hydrolyzes alpha glycosidase but not beta glycosidase, and whereas emulsion hydrolyzes the beta glycosidase but not the alpha glycosides. It should be noted that a given enzyme can exhibit more than one of the specificities. And for example, esterases hydrolyze only esters, may also hydrolyze one enantiomer and more rapidly than the other. Now, the factors responsible for enzyme specificity. There are certain factors which are responsible for enzyme specificity. These are: an enzyme must recognize some common structural features on substrate as well as coenzyme for specific catalysis. Catalysis should take place at a specific region on substrate or coenzyme, and stereochemical consequence should be enzyme controlled. For example, this is the NADH and this is attacking on the ketonic group. Here the ketonic group is alcohol dehydrogenase hydrogenase enzyme is acting here. It reduces the ketones to alcohol with the distereotopic hydrogens. Carbonyl group of ketone has two faces and is therefore enantiotropic. In presence of enzyme, coenzyme NADH releases HA from the rear side of keto group, as a result of which methyl hydrogens become diastereotopic. In this example, methyl hydrogens cannot be distinguished and therefore become homotopic. Now, proximity effect and orientation effects are very important for catalytic activity. Generally speaking, there are four important factors for the specificity of enzymes. The first one is the chemical apparatus, the second one is the binding site, And the third one is the correct orientation of substrate. Chemical apparatus, there must be a chemical apparatus which can polarize the bond of substrate. Binding site, binding site can immobilize the substrate in correct geometry relative to other reactive groups. Correct orientation of substrate, correct orientation of substrate, it permits each step of the reaction to proceed with minimal translational and rotational movement about the bond of substrate. The fourth one is the mode of fixation and mode of fixation of substrate. And this should lower the energy of activation of enzyme substrate complex. Proper distribution of charge on active center and its geometry are responsible for the decrease in overall entropy of the transition state. Now, the next very specific characteristic of the enzyme is the catalytic power. Enzymes catalyze rates of biological processes at extremely faster rates. A chemical reaction in presence of enzyme proceeds 100 to 1 million times faster, and these reactions occur at the body temperature and in the physiological pH range nearly equal to 7, and they exhibit their activity even when they have been extracted from the source. Third property is the regulation. In enzyme substrate reaction, the reaction velocity decreases as a function of time. Here, the reaction velocity is increased, but as the function of time, the reaction velocity gets limiting or it gets saturated. The decline in the velocity may be attributed to the following reasons: due to the approach to an equilibrium and associated influence of the reverse reaction, because there is reverse reaction is also happening and occurring, taking place because equilibrium is there, and the depletion of the substrate level due to occupancy of active sites, and substrate levels are also depleted, and the products of reaction may inhibit to enzyme or change them if the medium is not properly buffered. So, if the reaction is not buffered, medium is not buffered, then it will change the enzyme specificity, and also enzyme may be inhibited by the product's reaction. The enzymes may undergo some progressive inactivation at the temperature or the pH of the reaction due to instability. So, due to above reasons, enzymes are studied within their initial velocity. Initial velocity is the slope of tangent to the curve at the V0 concentration, and this concentration of enzyme also alters enzymatic activity. Now, the next factor is the enzyme. So, we know that the rate is equal to the product divided by time, and so the enzyme into time is equal to product. If the substrate concentration is initially very high, the rate will first follow a zero order course, and later on it will fall down to the first order kinetics. So first it will follow the zero order kinetics, after that then it will follow the first order kinetics, and the entire reaction is thus a mixture which cannot be described by a single reaction order. We know that the rate is equal to product divided by time, and hence enzyme into time is equal to product. This property of enzyme was described for many years as enzyme time product relationship. And with the given concentration of enzyme, a point may be reached when all the substrate molecules get bound to the enzymes and further increase the increase in enzyme, which does not have any effect on the formation of the product. So here enzyme concentration is increasing, so enzyme activity is also increasing, and but after enzyme activity is getting limiting, okay, limiting value. Here the initial velocity is there and concentration of substrate is there. As we increase the concentration of substrate, initial velocity increases, but as we increase more and more substrate concentration, but the initial velocity does not increases and it becomes limiting. Concentration of substrate, as already stated, increases initial velocity and it is depicted in this figure. The fourth one is the effect of temperature. The actual temperature at which enzymes show maximum activity depends on a number of factors like purity of enzyme, presence or absence of activators on inhibitors, and it also depends upon the method which are followed during the course of reaction. And the effect of temperature on velocity of a large number of chemical reactions is according to the Arrhenius equation, and which is as d ln k upon dt is equal to ERT square E upon RT square, where E is the energy of activation, T is the absolute temperature, K is the velocity constant, and dt is the change in temperature. In most of the reactions, they follow Arrhenius equation and over physiological temperatures. When some reaction is allowed to occur in presence of enzymes and inorganic catalysis, enzymatic reaction has lower activation energy. And an example of the effect of temperature on velocity of enzymatic dephosphorylation of ATP is some given, and it gives some, besides some time temperature changes or increases also results in the inactivation of enzymes. Enzymatic instability is sometimes to the range of 30 degrees centigrade, and most of the enzymes get denatured at 60 degree centigrade. Activation energy for the inactivation of enzymes is quite high, of the order of 40,000 to 1 million calories, 1 lakh calories per mole. However, trypsin has considerable ability to recover from inactivation. Each enzyme show maximum activity at certain temperature, and which is known as optimum temperature. Here it is the optimum temperature is shown. This is the initial velocity, and the maximum velocity is achieved here at the temperature 50 degrees centigrade, and it is known as here optimum temperature. So most of the enzyme has maximum activity. Here it is the effect of temperature on the enzymatic dephosphorylation of ATP. How the log values are getting decreased on increasing temperature. Here this is the relationship. Now the fifth one is the effect of pH. For the effectiveness of each enzyme, there is also an optimum pH, and the optimum pH for most of the enzyme is in the pH range 7 to 8. Here this is the initial velocity which increases, and its maximum value is at near about 7 pH, and this is the optimum pH. Crickwood gave a theory regarding effect of pH on reaction rate, and which is based upon the fluctuation forces between the protein molecule, that is the enzymes, resulting from the protonic charge. It has been found that trypsin and chymotrypsin follow it that only a single ionizable group is involved in the activity of enzymes. Activity loss of enzymes at extremely low or extremely high concentration may be due to pH induced conformational changes. The sixth factor is the energetics. The total functioning of animal and plant body is the result of constant supply of energy from the outside source in the form of food stuff. The chemical reactions involved in the constant flow of energy are enzyme catalyzed. Principles of energetics are helpful in knowing if a particular biochemical reaction is thermodynamically physical or not in presence or absence of enzymes. Energetic properties are useful in giving information about the reaction pathway. The enzymes in dilute solutions are helpful in attaining equilibrium at faster rate as directed by law of thermodynamics. and enzyme cannot catalyze the energetically unfavorable reaction. Enzyme cannot. So these are the few examples. For example, mono debenzylation on adenylic acid phosphate will give a branched ATP of this type of structure, and this is the branched type of ATP, and this is the isomer of adenosine triphosphate. Now, the next one is the binding activity of enzymes and redox potential. A system with slightly lower potential than the other doesn't mean that it cannot oxidize later. The driving force of reaction depends upon the actual concentration or activities of the reactants. They may effectively overcome an unfavorable potential difference. For example, this is the reaction of ethanol and diphosphopyridine nucleotide, and it gets in equilibrium with the acetaldehyde, that is the product acetaldehyde and diphosphopyridine nucleotide plus H ion. In this reaction, alcohol dehydrogenase can combine more tightly with reduced rather than with oxidized coenzyme. So, shift in potential through coupling with protein may be a technique used by nature to overbrace high potential groups. The next factor is the relation with precursors. Relation with precursors. Zymosins are the enzyme precursors. Comparison of zymosin with enzymes indicates that it is possible to determine what changes are desirable in order to develop enzyme activity. So, this is the very important factor. The ninth factor is the activation or deactivation by ions. Some enzymes become active in the presence of cations such as magnesium ion, calcium ion, magnesium ion, zinc ion, sodium ion, or potassium ion. And some chloride ions enhance the activity of salivary amylase in our saliva, which the amylase is present, and this is enhanced by chloride ions. And concentration of ions also influences enzyme activity. 10th factor is the impact of inhibitors. Inhibitors also gives influence the specificity of the enzymes. Inhibitors are the compounds which decrease the rate of enzyme catalysis reactions. They may be reversible as well as irreversible, and reversible are covalently bounded to the enzymes, and there is equilibrium between free enzymes and the enzyme bounded with inhibitors. Irreversible inhibitors render the enzyme permanently inactive. So this is the enzyme, this is the inhibitor, which is this inhibitor blocks the enzyme's active sites, and this makes the enzyme inhibitor complex. So here when these are the all the factors which influence the enzyme specificity. Now the this lecture is stopped here, and thank you.